Great. Thank you, Martin, for the invitation, and thanks everyone for coming out to the first session here. Today I'll be talking about some of the technologies and platforms that we work on in my group. Most single-cell work currently in the field really focuses on gene expression, and you saw some beautiful work presented by John in the first talk. And there are a lot of reasons behind that, right? You've got gene expression and exploring the transcriptome is highly informative for cell state or cell type. It's relatively easy to access. You've got this really nice poly A tail that's kind of this molecular tag you can latch onto and target those molecules specifically. And it's fairly readily interpretable, right? You're working in genes, and as geneticists, we typically think in terms of genes, and we know what most genes do. Not all, and that's a whole other topic, but it's a little easier to wrap your mind around the gene space. But there's a lot of other things going on, right? You've got all of the regulatory landscape that's driving that gene expression, or other regulatory aspects that might not be directly detectable or result in gene expression changes. The topic that my group focuses on the most is really looking at these regulatory sequences. So these are regulatory elements in the genome. They're defined typically by accessible chromatin. So these are regions where there's no nucleosome bound at the region of the genome. So it makes that DNA actually open and accessible to the transcriptional machinery or other DNA binding proteins. So these are kind of the four areas that I'm going to be talking about. Are the platform of what we use to assess this. A couple of vignettes that are both unpublished about applying it to corticogenesis, and then how we're starting to try to incorporate spatial information to our assay. And then I'll touch briefly on some other strategies where we've extended the platform to a lot of other different properties. So the first is this SkyAttackSeq workflow. I like to use figures from this paper from 2016 from the Greenleaf and Chang labs at Stanford to really motivate looking at AttackSeq as a mark for cell type identity. And what they did was they took flow-sorted populations based on cell surface markers from a number of different blood lineages, and then performed RNA-seq or AttackSeq on these samples. And what they were able to show is that in using AttackSeq to partition out these cell types, it did a better job at getting the cell type purity. So there's more information to discriminate between the cell types. And if you think about it, it kind of makes sense for several reasons. One, RNA transcription is an analog signal with a high dynamic range. The difference between two cell types might be fairly subtle. So you might have 500 transcripts per million versus 600 transcripts per million for a gene between two different cell types. And that's not a huge difference. Whereas with regulatory element usage, it's close to binary. We do use counts, but it's typically where these regulatory elements are either on or off. Not only that, but there's a lot more, there are a lot more regulatory elements in the genome than there are genes. So there's over 2 million annotated regulatory elements in the genome versus only tens of thousands of genes. So there's a lot more signal that you can go off of. They further broke this down, showing that distal elements do the best. And this kind of makes sense. For one, there's a lot more distal elements than promoters. But if a gene's on or active at all, the promoter is going to be accessible. And in fact, as I'll touch on a bit later, the promoter accessibility, the amount of chromatin accessibility at a promoter for a gene doesn't correlate very well with expression. It's generally going to be open and accessible if the gene's on at all. So the way we access this is using a transposase-based strategy. So you can use this hyperactive transposase loaded with sequencing adapters. And if you incubate this with regular purified genomic DNA, you get a genome sequencing library. So it fragments the DNA into sequenceable sizes and appends adapters in a single step. Jason Buenrostro back in 2013 showed that the concepts behind DNase hypersensitivity mapping, which is a physical constraint of being able to use enzymes on only open regions of chromatin, also works for this transposase-based library preparation. So if you expose your native chromatin to this transposase reagent, the transposase can only act and insert sequencing adapters and fragment DNA in these open regions of chromatin. So when you sequence this created sequencing library, you only get reads from these open regions, and you can call these peaks as these accessible regions in the genome or active regulatory elements. So how do we take this to the single cell level? There's several strategies out there. I want to point out there's a great paper from the Teichmann lab on being able to do single cell, single well, single cell AttackSeq that came out in Nature Communications. It's a great protocol. It works really well if you have low cell numbers. There's some strategies that were designed early on using microfluidics chips. More recently, there's commercially available platforms, 10x Genomics and Bio-Rad, both have papers that are, at least preprints that are out on their platforms, as well as some microwell-based strategies, but that I'll touch on a little bit at the end. But the strategy we use for the most part is single-cell combinatorial indexing. And we think it has some advantages. Namely, there's combinatorial scaling of throughput, meaning the more cells you profile, the cheaper it is per cell. So it's this nonlinear cost expansion of increasing cell numbers. It doesn't require any special equipment. It's adaptable to a variety of properties, and it's quite inexpensive to perform. So the way this works is you can take your cell culture, your tissue sample, whatever, and the first step is isolating nuclei, which has some advantages. Namely, if you're dissociating cells that are alive and viable, often they're changing their regulatory landscape or their transcriptome during that isolation process, which is a variable you really don't want to introduce. So killing them and isolating the nuclei right away is kind of a nice strategy to kind of lock them in place. And then we perform our in situ tagmentation, so the transposase can actually enter into intact nuclei and act on chromatin within the nucleus and create your library there. And the enzyme actually stays tightly bound and kind of glues those newly created sequencing molecules in place and traps them in that nucleus. But we don't just use regular sequencing adapters, we actually have DNA barcodes on these adapters. And these barcodes correspond to wells of one or more 96 well plates, such that all of the nuclei within the same well are all going to have adapters with the same barcode. Now, if I pool all of those nuclei together and were to reach into that pool and grab a random nucleus, it would have a random one of those 96 barcodes, right? If I were to reach in and grab a second one, there's only a 1 in 96 chance it has the same barcode that I just pulled out, right? So we leverage that probability to do a limited number of nuclei based on the number of 96 well plates we do at the transposition stage, such that as we sort these into new wells of plates, there's a low probability of those nuclei having the same barcode, so we can tell those library molecules apart. We then perform PCR with another round of indexing, such that we can then pool everything together and perform sequencing. So just in this toy example, you can see there's, in this 4x4 barcode situation, we get around a dozen or so uniquely barcoded nuclei. So when applied to the 96 well plate scale or 384 well plate scale, we can get thousands or tens of thousands of single cells without having to do large numbers of plates. So the first little vignette I want to talk about is applying this to in vitro corticogenesis. And this is available as a preprint as of about a month ago. And the first aspect of this was actually some improvements to our workflow. So one of the things we noticed when imaging nuclei that were exposed to transposase, the transposase here is shown in red and DNA is in DAPI in blue. You can see that the transposase doesn't penetrate very far into the nucleus, and we weren't happy about that because we know there's active chromatin in there that's not getting transposed. So we started using this reagent. It's marketed as Pitstop 2. What it does is actually opens up the nuclear pore, and this enables more transposase to actually enter in. There was kind of a roadblock that was happening before where all this transposase was kind of getting gummed up in the nuclear pore, and now it enters more freely, and we get a lot more transposase entering deeper into the nuclear core. And we can quantify that based on imaging. We also did a bunch of flow sorting of nuclei showing that we get increased transposase fluorescence. And this translates directly to a greater number of unique reads per cell. So we're getting more information that we can obtain of accessible chromatin per cell, and it ranges between about 2 to 3-fold on the lower end, up to as much as 8-fold more unique reads per cell. So we applied this to an induced pluripotent stem cell derived mini brain model. So this is using the spin omega protocol, and it's a collaboration with Brian O'Rourke, also at OHSU. Where we took these iPSC cells and differentiated them into these cortical organoids, forebrain cortical organoids. And you can see some imaging here done by Brooke DeRosa, a postdoc in Brian's lab. And you can look at the preprint. There's lots of pages of this. I don't know the imaging as well, but they look nice, and there's nice rosettes of cortical layers. And then we performed our single-cell ATAC-seq workflow. And then from there, we used the data to look at either local chromatin accessibility networks or the global accessible chromatin landscape, different transcription factor motifs being accessible throughout the genome. And just to show the comparison of organoids that were prepared without using this pit stop reagent and ones that were using the pit stop reagent, and with pit stop is in red here. This is a log 10 scale on the x-axis, and you can see there's a substantial increase in the number of unique reads that can be obtained per cell. And this persists with equal sampling of read depth. So we then take all of our reads, align them to the genome, call our peaks, and we create this matrix of the amount of signal that we have at each regulatory element for each one of our cells. We used to use a strategy called latent semantic indexing to kind of normalize out. As you can tell from the previous slide, there's a pretty big dynamic range, or range in the amount of unique reads we're sampling per cell. So latent semantic indexing could help with normalizing that essentially. But recently we've been using this new strategy from Stein Arts Lab that I really want to plug because it's an awesome tool called systopic that does an excellent job of actually pulling out something kind of similar to eigengenes that John described, but these topics of co-accessible loci in the genome. So it's a way of reducing the dimensionality of your data in a way that does a really good job at actually capturing some of the biological themes of the data. So we can take that and then put it into UMAP, very similar to t-SNE, so much like what John described, where each one of these points is a single cell. There's something 12,000 or so here. And they're positioned more closely in two-dimensional space based on how similar their profiles are. And then we perform cluster calling using a Louvain-based strategy. And if you split it by the organoid, or more interestingly, days in vitro, we can kind of get a sense of the progression here. As well as whether there's pit stop 2 treatment or not, and we observed very little, or essentially no difference there. We have additional data supporting that this pit stop 2 reagent doesn't actually change the chromatin landscape. So we can then take our clusters and do something very similar to what you do with RNA, where you look at differentially expressed genes, and we look at differentially accessible regulatory elements between these different clusters. And we can take those regions and then do motif calling to look and see what are the transcription factors that are likely driving these changes in chromatin accessibility. And the transcription factor programs and networks in corticogenesis and neurodevelopment are pretty well known, so it was actually pretty easy for us to define which each of these clusters are. So 1 and 2 are these early neural progenitors. 3 are more outer radial glia that then transition into 4, which are these intermediate progenitors, and then 5 are these full glutamatergic neurons based on these motif accessibilities in these differentially accessible loci. We can also look at the local level, so looking at specific factors that we know drive these processes. For example, SOX2 here is known to be highly active in the earlier stages in neural progenitor cells, and you can see around the region here, each one of the rows is a single cell. There's a lot of accessibility signal at the regulatory element surrounding SOX2 that decreases as you move through differentiation. Similarly, one that are active later, like NeuroD2, there's hardly any accessibility at the regions around the gene earlier on, and it's not until you're in these more full glutamatergic neurons where you're getting this NeuroD2 expression or accessibility around the gene. We can also do this pseudotemporal ordering. So this is actually walking a path through our network of cells. It looks kind of messy here, actually, because it's done in 3D, but unfortunately paper is 2 dimensions, so if you want to have a plot, you typically have to do it in 2 dimensions in a journal, so it's kind of squashed into 2, but that's why the wireframe kind of looks a little messy. But we can use that to then walk these paths through the differentiating cells through this process and look at things like topics through this pseudotime, through the differentiation progression, or different branching events throughout the neuronal lineages. We can also look at the accessibility through pseudotime. So this is accessibility of motifs throughout the genome for different transcription factor motifs. For instance, the SOX genes are in this cluster here where you've got higher accessibility throughout the genome earlier on, then it decreases later. So this is looking at the global effect of the transcription factors. And then we can also go back to the local level using a tool called Cicero from Cole Trapnell's group that we love. It does a really good job. As I mentioned before, the promoter accessibility doesn't correlate well with gene expression. But what does correlate well is the signal at enhancers that are linked to that promoter. So if you aggregate the signal across all of these enhancers that are associated with the promoter for a gene, you can actually get a fairly reasonable assessment of the gene activity score that correlates fairly well with gene expression. So we can essentially infer gene expression using these models. And get a similar plot that we had for transcription factor motif accessibility, but this time it's actually looking at the activity around the gene that transcribes that transcription factor. And these are specific transcription factors, but we also get it for all genes in the genome. And when we put these two together, we can start to build out some of these networks in neuronal development. So we decided to focus on this REST story, which is a known repressor of neuronal differentiation. And what I'm showing you here is through pseudotime on the x-axis, you've got these different colors. So the filled in lines are the global motif accessibility for that transcription factor. And then the dashed lines are inferred gene activity scores for those factors, based on the local chromatin architecture. So you can see that the gene activity of REST goes up as it's repressing differentiation of neurons during radial glia stages. And the gene activity score is up, but then the subsequent global effect of that gene activity results in the motifs for REST being opened across the genome. And then it subsequently decreases as you shift into intermediate progenitors and then full-on glutamatergic neurons. So we're able to start to dissect out some of these networks. The other little vignette I want to talk about is also unpublished work on trying to incorporate spatial information into this assay. And it's actually a really simple, almost kind of dumb approach, but it's actually using just microbiopsy punches, so just doing physical isolation of small regions of tissue. And this came about from actually, we were trying to figure out this really sophisticated strategy, and then we talked to actual biologists who know what's going on at that level, and we asked, like, what's the resolution that you actually need? And we realized we could actually achieve that using a much more simple approach. So this actually isn't drawn to scale. This is from early grants, but the strategy is still the same, where we take alternating slices of thick and thin tissue slices. The thin ones are used for imaging, so around 12 microns is typically what we'll do for that. And then we'll take a slice that's between 100 to 300 microns in thickness and lay that on a slide, and then actually do microbiopsy punches of that and drop each of those into wells of a 96-well plate. So it's just doing multi-regional sampling, but with pretty small regions. And what's nice about our combinatorial indexing assay is we can then use our transposase barcodes to actually track what punch each of these cells came from. So we actually use that as sample encoding, where it's a really great strategy for multiplexing lots of different samples, which is what we have here. We did a lot of work, specifically Casey in my lab, optimizing getting nuclei isolation in high quality from these microbiopsy punches, including different washes that actually improve the data quality substantially. We also get nice imaging from the slides adjacent, so we just wanted to make sure that that works. And then we also have been experimenting with different punch sizes as well as section thicknesses. The only commercially available punches, they only go down to 250 microns, so we're actually fabricating some that are 100 microns, because one of the things we found is we get better efficiency with smaller punches, so I think we can get higher and higher resolution. And that's kind of illustrated here, where the filled in dots that are more to the left here are the 250 micron punches. These are 500 micron punches, and this is the x-axis is punch volume, and y-axis is the efficiency, essentially, the number of nuclei we get per cubic micron of tissue. And you can see it's just a lot more efficient with these smaller punches. And the actual section depth doesn't make a huge difference either. So we think we can get a little higher resolution. And just to give you an example of one of the experiments we did, we took 6 slices throughout the mouse somatosensory cortex, and we did 6 punches from each one of these slices, 2 from upper cortical layers, 2 from lower cortical layers, and then 2 from regions of white matter. Put them in a plate, dissociated them, and then we actually get enough nuclei to split that into multiple transposase plates, such that we get quadruplicate barcodes for each one of these punch samples. And then pooled everything, did a redistribution into 2 PCR plates. We get around 90 single cells per punch, which corresponded to around 8,000, close to 8,000 cells, where we have a threshold of over 1,000 unique nuclear reads per cell that are actually in peaks, so a reasonable threshold there. And instead of color coding them by punch, which is 96 different colors and just looks like confetti, I split it into whether the punches are derived from upper or lower cortical layers versus other, these white matter regions. And what's pretty apparent is you see this one cluster up here in the right corner where it's you really don't get any cells that are from the other punches, and which kind of makes sense. These are the cortical regions, so those are the neurons, right? We also wanted to show that we can integrate this data with other experiments. So we have a 384 well punch set, and you can see the cells cluster in all of the different regions, as well as the upper, lower, and other. We also compared it to whole brain samples, whether fresh or frozen, and we actually get very, very little sample variability from the fresh or frozen or where the cells are from, or using this punch assay. We can then look at global accessibility for different factors. So for example here, SOX9 for astrocytes is highly active in this cluster, so those are astrocytes, as well as activity or accessibility of different neuronal transcription factor motifs, or look at local regions. So, for instance, microglia have high accessibility around C1, QA, and C, and you can see that highlighted here with the cells in the microglia cluster with a lot of accessibility. Similarly, oligodendrocytes and with MOBP, there's this nice boost in accessibility around those genes for marker genes. And then we can start to dissect out some of this spatial information, like looking at these regions, you can see they're about 50-50 neuronal versus non-neuronal in these upper cortical layers, whereas if you move into these other regions, it's almost exclusively non-neuronal cells, which makes sense, because they're these white matter regions. And we can look at it actually through this, the spatial gradient through the slices, where as we move further back or throughout the tissue, we get pretty consistent levels of about 50% neuronal versus non-neuronal, as well as the high shift with a little bit of a bump earlier on in the punches in the other region. So the last thing I want to talk about is how we've extended this assay to actually look at some other properties. So here's a subset of some of the published work that's a collaboration between my group, Jay Chendre's group, and Cole Trapnell's group, as well as Illumina, on expanding out this single-cell combinatorial indexing workflow space. And just to sum it up kind of on the epigenetic landscape type chart, the ones in green are published with several strategies here, and some we're working on currently to look at regulatory proteins and histone modifications. To highlight a couple of these, and this is published work, so I'm going to go through it fairly quickly. We did a single-cell whole genome sequencing strategy, where the key to this was actually, instead of getting the transposase to insert only at active regions and open regions of chromatin, we actually found a way to open up nucleosomes and remove them to enable the transposase to insert everywhere in the genome, so we can get single-cell whole genome sequencing using the same strategy. We applied it to primary stage 3 pancreatic ductal adenocarcinoma. We're able to identify 3 different subclones of the tumor based on their copy number profiles. And then since then, we've extended this. This is work led by Yi Yin. This is also on bioRxiv with the Sky-Leonti approach. So it's a way of kind of boosting the signal that we get per cell using an in vitro transcription step that gives us more unique reads per cell and better resolution at copy number calling. We also have some work that's published, and I'll touch a little bit on some unpublished advances on that, is a strategy, now that we've been able to open up the genome, like I said, for the single-cell DNA sequencing, we're able to look at genome-wide properties such as single-cell DNA methylation. So that's with this SkyMet assay, we're able to show that we're able to look at primary mouse cortex and dissociate and identify different cell types that are present. I will say single-cell methylation is excessively expensive. The cost to prepare each cell is very cheap using our strategy, but the cost in sequencing is just huge, because you're looking at genome-wide. You need millions of reads per cell as opposed to tens of thousands or hundreds of thousands with ATAC-seq, right? So some of the new advances on this that we haven't published yet are incorporating strategies to do artificial methylation at regions of open chromatin. So this is a strategy several groups have used, where you use a GPC methyltransferase to methylate regions of open chromatin in this GC dinucleotide context. So then you can dissociate or pull out those 2 signals. And in blue here, I'm showing CG methylation percentage. So this is the standard native DNA methylation that wasn't artificially introduced. And you can see that if we center our focus window around CTCF motifs in the genome, there's this nice decrease in methylation around the CTCF binding site, which is known because it's usually hypomethylated at these binding sites. So we get that nice drop with that blue line. And then this green line is our chromatin accessibility signal. So you can see that there's this spikiness. These are the regions in between nucleosomes, so we get a nice nucleosome positioning signal. And then around CTCF, you see this big spike, and then a drop at the middle, and this is actually the footprint of CTCF at that binding site so that we're able to capture. In the last minute, I'll talk about some of the next extensions we have with this. So I want to highlight this preprint from Jason Von Roester's lab. He did an excellent job doing droplet-based single-cell ATAC-seq. where he actually did combinatorial indexing in droplets. My group was trying to get droplet-based ATAC-seq to work for like 2.5 years, and turns out we just weren't really good at purifying oil. And so we kind of gave up on that and we started shifting to physical compartmentalization strategies. So we're doing some work on the Takara isolate system, as well as the Celsy system, which I'm showing here, which is a micro-well-based platform where you get these chips that are between 100,000, there's 100,000 micro-well chips, 250,000, and then million micro-well chips. And we developed the ATAC-seq workflow actually on this platform using kind of their open tool development platform. And just to highlight, you can see that we get nice signal at these accessible regions. This was actually the first shot at attempting this single-cell ATAC-seq workflow on the Celsy platform, and it worked fairly well. Now we're in the process of iterating and optimizing every single step in the workflow so that we can release it, and so we're really excited about that. We think it's going to dramatically increase the throughput that we can obtain from each of these experiments, and moving into the million-cell data set space pretty quickly per prep. With that, I want to acknowledge my lab collaborators and funding sources, as well as my lab and the people who actually did all the work that I showed you. So thank you, and I'll take any questions.
Thank you.